Bismillah ar-Rahman ar-Rahim. This is the third part in the revision of histological slides for the second year. The first group in this revision is the compound slides. We have 5 compound slides. We have duodenum and pancreas, we have testis and epididymis, we have human liver and gallbladder, we have thyroid and parathyroid and the spermatic cord. The duodenum is a luminal organ that comes always with the pancreas, as it is closely related to it. We have lobules of the pancreas separated by these white spaces due to the dissolving and disruption of the thin, fine connective tissue septa present in the pancreas. And here in the duodenum, you will see the villi and crypts, and the most important is the presence of the Brunner's glands. And this is, of course, the musculus. So, look for the Brunner glands and look for the associated pancreas. This is the duodenum and pancreas. The second is the human liver and gallbladder. It may come on any shape in any piece, but it is always accompanied with the gallbladder, which is a hollow organ. When you look at the slide under the microscope, you will notice that the human liver is characterized by demarcated trabeculae, and you will notice only the central veins, but no hexagonal lobules can appear due to the thin connective tissue trabeculae. And beside it, you will see the wall of the gallbladder, which is thin and characterized by the folding of the mucosa. Also here, this is the liver. We have the central veins, no landmarks for the hexagonal lobules, and this is the folding of the mucosa of the hollow organ accompanying this liver tissue. And this is the spermatic cord, very easy to be diagnosed because it contains the vas deferens, the characteristic small hollow lumen with the bulky musculosa here. And this is the actual picture of your slides under the microscope. Here we have the musculosa, the lining epithelium of the vas deferens, which is pseudostratified columnar epithelium, and beside it you will notice the bundles of the skeletal muscle fibers at one side, and at the other side you will notice blood vessels. These are the pampiniform plexus of veins, and these are the cremasteric muscles. So, the vas deferens is accompanied by 2 structures, and they are all covered by thin connective tissue. One of these structures is the pampiniform plexus of veins, and these are, and here, and the other is the skeletal muscle, involuntary cremasteric muscle, as you see here and there. This slide is also a compound slide. It is for the thyroid and the parathyroid. The thyroid is the big mass, more lightly stained compared to the small dark. There may be 1 or 2 parathyroid masses accompanying the section of the thyroid. By looking under the microscope, it is easily diagnosed, as the thyroid follicles are very easy. They are filled with the colloid pink material inside, and the parathyroid, as you all know, are formed mainly of basophilic cells, which are the principal cells. The other compound slide is the testis and the epididymis. The color has no significance. They may come in the blue color, both of them, or red color, both of them. This depends on the staining. And of course, the big mass is formed of tubules, which has a thick wall and relatively narrower lumen, while the small mass is formed of wider tubules with a thinner wall, which is the epididymis. By looking at the testis, the big mass, under the microscope, you will notice immediately the seminiferous tubules, and inside you will see the fine hairs of the sperms. While looking at the small mass, you will see the lining epithelium of the epididymis, pseudostratified columnar, ciliated with non-motile cilia or stereocilia, and inside we have the sperms inside this wide lumen. The second group of slides is the lobulated slides. We have 6 lobulated slides. We have the pituitary, kidney, human liver, prostate, pancreas, parotid. Type. We will start by the pituitary and the kidney. Both of them are formed of 2 lobes or 2 areas. We have a dark area and a pale area. In the pituitary as well as in the kidney, we have the dark cortex and the paler medulla. This is the pituitary gland. It is a small structure, and you can see that it is formed of 2 lobes, the anterior lobe, which is darkly stained compared to the posterior lobe or pars nervosa. Sometimes it forms like a C-shape, or it completely surrounds the anterior pituitary, completely surrounds the posterior pituitary. Here you see this C-shaped of the pars distalis, and this narrow area which is darkly stained and closely related to the pars nervosa, it is the pars intermedia, and the white area, it is the remains of the lumen of the Rask pouch from which the anterior pituitary arise. Here we have the anterior pituitary, and this is the posterior pituitary. Anterior is darkly stained compared to the posterior, and the structure is very easy to be diagnosed by its small size structure and the appearance of this distribution of colors of posterior pituitary containing the pituicytes, and here we have the pars intermedia on the surface, and then the narrow cleft here, and the pars distalis. Here the kidney is much bigger than the pituitary in size, and you will see that it is formed of the outer cortex, which is darkly stained compared to the medulla. Here you can see now under the microscope that by looking at the cortex, you will notice immediately the very important and characteristic structure, which are the glomeruli. These glomeruli are highly scattered in the cortex. By going deeper towards the medulla, you will notice the collecting tubules and ducts, and of course you can see the start of the calyx lined by transitional epithelium, but this is very important, and it is enough to diagnose the kidney by diagnosing the glomeruli as well as the collecting duct. A very easy lobulated structure is the liver of pig. Any mass can come triangular, rectangular. However, under the microscope, the picture is very characteristic. You can see the hexagonal lobule due to the presence of these trabeculae, and inside you will notice the central vein and the cords of the liver cells radiating from this central vein towards the periphery. Again, central vein and the cords of the liver cells. Hexagonal lobules are very evident in this lobulated slide, which is liver of pig. You have to mention the species because you have another slide for the human liver. So in this slide, you have to mention that it is liver of pig. Again, lobulated slide, which is also very easy, it is the prostate. Here the prostate is very characterized by the presence of the prostatic urethra. Here the prostatic urethra in the middle of this organ, which is divided into lobules by this red connective tissue septa. And inside the lobules, you will notice the prostatic acini, very small in size, relatively darkly stained. As you see, these are acini in one of the lobule, another lobule with acini, and so on. And the most important thing is the presence of this urethra inside this tissue, with the characteristic branching as you see in this picture, curved like this picture. So this is the prostate. Another lobulated slide, which is the pancreas. The pancreas very similar in appearance to the parotid, but you will compare between them. As you see here, the septa are very fine, so they are cut, leaving white spaces, but the septa in the parotid are thick and they are still present and stained by the red coloration. The most important than this thing is the appearance of the lobules under the microscope. Under the microscope, look at the lobule. You will not notice any duct inside the lobule, which is the intralobular duct. But you will see ducts in between, of course. But inside the lobule, you will not see any intralobular ducts. You may be lucky to see also these lightly stained patches, which are islets of Langerhans. As you see here, I will make it more bigger. You will see that we have these dots of the islet cells are scattered, and these are the acini, taking the shape of the rosettes with a dark peripheral part or a basophilic peripheral part and acidophilic central part. Inside the lobule, I cannot see any duct, but I can normally see ducts in the connective tissue in between the lobules, which are the interlobular ducts and interlobar ducts. Also in this picture, here you will see the lobules formed of the acini, and this is the islet of Langerhans. No intralobular ducts. Also the lobules, no intralobular ducts, but only islets of Langerhans, but not necessary to find islets in your section. The most important thing is the absence of the intralobular ducts. The second slide, which is the parotid, and I have mentioned how they differ in the appearance of the connective tissue septa in between, but the most important thing is the presence of the ducts inside the lobule. This is the lobule. Look at the lobule, you will notice that it has ducts. And this picture is from your slides, not from the internet. This is the slide on which you will be examined, and these are the ducts inside the lobules. This and this and this and this and this. Inside, look at the lobule. Don't look at the connective tissue in between. We will look for the intralobular ducts or the secretory ducts, which is very characteristic for the parotid and absent from the pancreas. Also look for this picture. Here, very nice. You will see this is the lobule. Here, I have a duct, a duct, a duct, a duct. Don't look for the ducts in between the lobules. You will look for the intralobular ducts inside the lobule itself. This is a lobule, and this is a duct inside. Now, this is a group of the oval structures. The oval structures, we have only two oval structures, the suprarenal gland and the ovary. The suprarenal gland is characterized very much by the appearance of the zona glomerulosa, which is very pale in staining or pale basophilic, and then we have the rest of the cortex and the medulla darkly stained inside, while the ovary is characterized by the appearance of the follicles inside this tissue. Now, look for this oval structure, and this oval structure, not necessarily so oval, but the most important thing is the appearance of this zona glomerulosa. Now I can make more bigger, and here you can see the zona glomerulosa, the arches of these pale basophilic cells. And these are the fasciculata, reticularis, and then in the medulla, characterized by the presence of these blood vessels, slightly darkly stained compared to the cortex. Notice here the zona glomerulosa, also very nice. And the fasciculata, reticularis, and the medulla. Very characteristic is the zona glomerulosa. Also the other section, also for the suprarenal gland, but it is not so oval, يعني مختلفة شوية, مش rounded ولا oval, but the most important thing is the appearance of the zona glomerulosa, and this is the fasciculata, reticularis, and the medulla with the blood vessels inside. Now this is a picture under the microscope also showing the zona glomerulosa, how they appear, like the arches. Here, very characteristic. Please look for this layer, very important in this oval structure. And then the fasciculata, reticularis, and the medulla is in the center. Here is the ovary. As I have said before, it is characterized by the presence of the follicles all through the structure or the cortex of this structure. Look at the cortex, you will notice first the primordial follicles, the primary, the secondary, and larger follicles. Here also small follicles, primordial, and the different stages of the development of the ovarian follicles. Some may show the oocyte, some do not show the oocyte. This is according to the plane of the sectioning. All of the ovary slides should show these ovarian follicles. As you see, these are different slides from your slides, and all of them show the different stages. And of course, this one is the corpus luteum here, and this is corpus luteum. The center is the medulla, which is rich in the blood vessels. So, the slide of the ovary is very nice and very easy slide. As you see, all the slides are having the same feature, which is the appearance of the follicles at different stages of development, and in the center we have the medulla with blood vessels. Finally, we have the group of spongy structures, which are three structures only. We have the lung, of course spongy, but the lung is characterized by the presence of bronchioles and maybe bronchi, not necessary, and the submandibular gland characterized by the presence of the mucus acini together with the ducts of the gland, and the placenta characterized by the presence of the chorionic villi and the blood in between. And now we will see the three different slides. This is a slide, of course, it is spongy in appearance, so it is one of these three slides. If you look here, you will notice that it has a bronchus due to the presence of this cartilage here. And of course, this is a large blood vessels. Here you can see also this is a bronchus, and these are the alveoli. It may show only bronchioles like this also, not necessary to show the bronchi, but the presence of these empty alveoli with the bronchioles and blood vessels, which are very big, they demonstrate the lung tissue. Here the submandibular gland is different. Here you can see that we have these acini, but these acini are pale blue. They are not empty inside, not white inside like the alveoli. Also, you cannot see bronchioles. You can see only these ducts with no folding of the lumen, and these are blood vessels in between the lobules. Now you will see another slide. This slide, you will notice that it is characterized by these mucus acini, and these are blood vessels here, and this is a duct, and this is a duct. These are ducts and not bronchioles. The bronchioles are characterized by having the folded lumen. The third spongy structure is the placenta. Here we have the picture of the placenta under the microscope. Here we have the chorionic villi. Chorionic villi containing blood vessels inside, and also these are sections of chorionic villi of different sizes, smaller, larger, and in between the villi you may notice blood or not, but these chorionic villi are characterized in the large ones by the presence of this chorionic blood vessel, and these are the cells surrounding the blood vessels. These are different sections of the chorionic villi, Some are small as we see, and some are bigger, and they contain the blood capillaries. Also, you'll see other one here. These are the chorionic villi, and of course you notice that the chorionic villi are formed of blood vessels and cells. It is not empty. And here in between you will notice maybe there is blood, but in this section we don't have blood, so it is not necessary to have blood in between. But you will see that we have smaller sections of these chorionic villi. We have big sections, small sections, and in between maybe an empty space or a space filled with the blood.